8th grade life science. DNA, the genetic material. Where are the instructions? How do your cells know what to do? Just like builders have blueprints to tell them how to build a house, your cells also have instructions. Your cells' instructions are molecules of DNA. What is DNA? DNA is the material that makes up our chromosomes and stores our genetic information. When you build a house, you need a blueprint, a set of instructions that tells you how to build it. The DNA is like the blueprint for living organisms. The genetic information is a set of instructions that tells your cells what to do. DNA is an abbreviation for deoxyribonucleic acid. As you may recall, nucleic acids are a type of macromolecule that store information. The deoxyribo part of the name refers to the name of the sugar that is contained in DNA, deoxyribose. DNA may provide the instructions to make up all living things, but it's actually a very simple molecule. DNA is made up of a very long chain of nucleotides. In fact, in you, the smallest DNA molecule has well over 20 million nucleotides. Nucleotides are composed of three main parts: a phosphate group, a 5-carbon sugar, and a nitrogen-containing base. The only difference between each nucleotide is the identity of the base. There are only 4 possible bases that make up each DNA nucleotide: adenine, guanine, thymine, and cytosine. The genetic code. The various sequences of the 4 nucleotide bases make up the genetic code of your cells. It may seem strange that there are only 4 letters of the DNA alphabet, but since your chromosomes contain millions of nucleotides, there are many different combinations possible with those 4 letters. But how do all these pieces fit together? James Watson and Francis Crick won the Nobel Prize in 1962 for piecing together the structures of DNA. Together with the work of Rosalind Franklin and Maurice Wilkins, they determined that DNA is made of 2 strands of nucleotides formed into a double helix, or a 2-stranded spiral, with the sugar and phosphate groups on the outside, and the paired bases connecting the 2 strands on the inside of the helix. The double helix of DNA. DNA's 3-dimensional structure is a double helix. The hydrogen bonds between the bases at the center of the helix hold the helix together. The bases in DNA do not pair randomly. When Erwin Chargaff looked closely at the bases in DNA, he noticed that the percentage of adenine in DNA always equaled the percentage of thymine, and the percentage of guanine always equaled the percentage of cytosine. Watson and Crick's model explained this result by suggesting that A always pairs with T and G always pairs with C in a DNA helix. Therefore, A and T and G and C are complementary bases, or base pairs. These base pairing rules state that A will always bind to T and G will always bind to C. For example, if one DNA strand reads ATGC, the other DNA strand will be made up of complementary bases. Hydrogen bonds hold the complementary bases together, with 2 bonds forming between the A and T and 3 bonds forming between the G and C. The chemical structure in DNA includes a chain of nucleotides consisting of a 5-carbon sugar, a phosphate group, and a nitrogen base. Notice how the sugar and phosphate group form the backbone of the DNA, the strands highlighted in pink, when the hydrogen bonds between the bases join the 2 strands. Okay, let's summarize. DNA stores the genetic information of the cell in a sequence of 4 bases: adenine, thymine, guanine, and cytosine. DNA is made of a long chain of nucleotides consisting of a 5-carbon sugar, a phosphate group, and a nitrogen-containing base.